Hello. Today we are getting into lesson 14. Now, I already went over part one with you, where we discussed the differences between RNA and DNA. And just quickly to review, DNA is deoxyribonucleic acid, and it is trapped inside of the nucleus of a cell. RNA is ribonucleic acid, and RNA can leave the nucleus. It also has a ribose sugar instead of deoxyribose. It still has the sugar phosphate backbone. And then RNA's nitrogenous bases are adenine, uracil, guanine, and cytosine. RNA is also single stranded, so it can leave the nucleus and enter the cytoplasm of the cell where proteins can be assembled. We already talked about the three different types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. We discussed how the messenger RNA molecule has been used to create some of the coronavirus vaccines that are out there. So that's very interesting, and you can watch that video if you haven't done so already. The big takeaways so far are that RNA can leave the nucleus, it's single stranded, uracil replaces thymine, and there's messenger, transfer, and ribosomal RNA. How do we put all of this together? Well, this unit is all about protein synthesis, and the word synthesis means to make, and we are making proteins. We've discussed the central dogma before. The central dogma basically states that information goes from DNA to RNA to proteins, and then proteins code for a trait. I've said it many times. It's a protein that codes for a trait. That's why my hair is brown. That's why your eyes might be blue, because of proteins. But how does that information go from the DNA that you inherited from your parents down to a protein that codes for a trait? And the interesting piece that gets that information from here to there is the RNA, and it happens during the process of protein synthesis, or the making of proteins. Recall from Unit 2 that amino acids are the building blocks of proteins. So, in other words, one amino acid is one Lego piece, and a protein is that big Lego castle. There is one kind of transfer RNA present for each of the 20 amino acids used in protein synthesis. So, one type of tRNA present for each of the different amino acids used in making proteins. To make proteins, we follow two steps: transcription and translation. Messenger RNA is made from DNA. Once it's created using the DNA as a template, the messenger RNA is going to leave the nucleus of the cell. It's going to find a ribosome. And once it finds that ribosome, transfer RNA will bring over amino acids one at a time. They'll link together, and voila, you have a protein or an amino acid chain. In order for cells to make proteins, the DNA code must first be transcribed. So, like we said, our two steps are transcription and translation. So, during transcription in the nucleus of the cell, the DNA that is coded, the message that is coded on the DNA, is going to be transcribed, which is a fancy word for copied, onto the strand of messenger RNA. Once that messenger RNA has been created, the code can leave the nucleus and go out into the cytoplasm looking for a ribosome. Here's another picture of this. You've got your nucleus right here. This is the outside of the nucleus, or the cytoplasm of the cell. Inside the nucleus, you have your DNA. The DNA is unzipped using RNA polymerase, and it's going to be used to create a strand of messenger RNA. As you can see in this graphic, anywhere that there would have been a T, it has now been replaced with a U. Uracil replaces thymine. Let's look closer at, well, that's the transcription process. So the first step, transcription and then translation. Once the messenger RNA leaves the nucleus, it needs to enter the cytoplasm and find a ribosome. That's what's taking place right here, and that's the second step of the process called translation. Once the messenger RNA leaves the nucleus, it finds a ribosome, and from there, ribosomal RNA, which is embedded in the ribosome, is going to alert the transfer RNA, which is carrying amino acids, to come over and drop off your amino acid. As the transfer RNA drops off its amino acids, they attach to each other, creating a protein. How does the transfer RNA know which one to drop off? Same as before, the rules for base pairing. At the ribosome, amino acids are linked together for specific proteins directed by the messenger RNA molecule. So if the messenger RNA brought with it AUG, the complementary amino acid or transfer RNA is methionine. The U would pair with an A pairs with a U, C pairs with a G. Peptide bonds link the amino acids together, forming a protein chain. Let's look at this again. Messenger RNA is created using the DNA. Once that message has been created, it leaves the nucleus and comes out into the cytoplasm to find a ribosome. At the ribosome, complementary transfer RNA are brought over to the messenger RNA and they drop off their amino acids. We call the messenger RNA side a codon. 3 nitrogenous bases on the messenger RNA side are called a codon. On the transfer RNA, the receiving side is called an anticodon. So the codons and the anticodons pair up with each other. That is protein synthesis in a nutshell. We're going to stop here and have you complete the protein synthesis draw along activity that's posted in Schoology. Make sure you post your drawing submission into Schoology for credit. I'd like you to watch this video of the Amoeba sisters making proteins, especially if you're still confused about anything that I went over. You're also going to read chapter 14, which is really going to clarify some things for you, and you'll complete the chapter 14 reading quiz. Thank you.